Poliovirus. What is it? It's a human enterovirus that causes poliomyelitis. It's highly contagious and can be life-threatening. It usually targets young children, but adults are susceptible as well. It's spherical, containing a single-stranded positive sense RNA, a capsid, which is a protein coat, and no envelope. Poliovirus can be transmitted by fecal or oral transmission. This could be from contaminated food and water, or from droplets in the air from an infected individual who sneezes or coughs. The virus moves into the GI tract. This virus binds to the epithelial cells of the intestines and releases its RNA within the cell. In the cytoplasm, it uses the cell's RNA polymerase to make copies of itself and uses the ribosomes to make viral proteins. This production of viral proteins and RNA creates new polioviruses, causing lysis and releasing newly formed viruses into the interstitial fluid to be transported by lymph nodes to the bloodstream. From the blood vessels, poliovirus can move into the brain and spinal cord and eat away at the motor neurons. In more advanced cases, this causes muscle atrophy, paralysis, difficulty swallowing and speaking, and even death. There are 3 types of paralytic poliomyelitis: spinal, bulbar, and bulbospinal. Spinal poliomyelitis refers to asymmetrical paralysis, usually of the legs. Bulbar poliomyelitis affects the brainstem that controls the motor nerves that function in speaking and swallowing. In bulbospinal poliomyelitis, the brainstem is also affected and disrupts the proper sending of signals down the diaphragm, a muscle important in respiration, which can make it very hard to breathe. The first documented outbreak of poliovirus in the United States of America was in 1894. President Franklin D. Roosevelt contracted poliovirus as an infant and suffered from post-polio syndrome, which means that polio affected him many years after his initial infection. Roosevelt was 39 when he suffered paralysis in his lower limbs, which made him wheelchair-bound. He was one of the founders of the March of Dimes, which was originally named the National Foundation for Infantile Paralysis. This foundation supported poliovirus research and treatment. So, how do we avoid contracting this dangerous disease? Well, the best way is to receive the polio vaccine. In 1955, Dr. Jonas E. Salk created the first injectable inactive poliovirus vaccine. In 1961, Dr. Albert B. Sabin created an active oral vaccine which was administered through the mouth. Throughout the years, the polio vaccine was further developed to be more functional. In 1994, poliovirus was no longer a public health problem. Because of this vaccine, many countries, including North America, have eradicated this virus, although its impacts are still felt in some countries across the globe.
Hi everyone! Today I'll be talking about SARS-CoV-2, the virus that caused the COVID-19 pandemic. We'll explore what makes the virus unique from a microbiology perspective, how it interacts with humans and the environment, and why it continues to matter today. SARS-CoV-2 is a coronavirus, a type of virus with a crown-like appearance under the microscope. It's an enveloped, positive sense, single-strand RNA virus. It emerged in late 2019 and spread globally, testing health systems, research efforts, and international collaboration. Studying it helps us prepare for future pandemics. This slide shows the key parts of the virus. The spike protein, shown here in red, attaches to the ACE2 receptor on human cells, allowing the virus to enter. That protein became the main target for vaccines. The virus also has membrane, envelope, and nucleocapsid proteins that help it replicate and stay stable. SARS-CoV-2 is genetically related to SARS-CoV-1 and a bat coronavirus called RATG13, which supports the idea that it came from bats, possibly through another animal before infecting humans. It's part of the same family as MERS and common cold viruses. The virus enters human cells using the ACE2 receptors. Once inside, it hijacks the cell's machinery to make more copies of itself. On surfaces, it can stay viable for hours or even days, depending on conditions. That's why early public health advice emphasized surface cleaning and ventilation. SARS-CoV-2 overwhelmed hospitals. Co-infections with bacteria or fungi, especially in the ICU patients, worsened outcomes. Overuse of antibiotics contributed to resistance. These infections made treating COVID-19 even more challenging. The virus spreads through respiratory droplets and aerosols. Asymptomatic transmission played a huge role in how fast it spread. People could pass it on without feeling sick. That's why public health strategies like mask wearing and social distancing became so essential. One of the biggest breakthroughs was the mRNA vaccine. It uses genetic instructions to teach the immune system to recognize the spike protein. This slide shows us a vaccine vial, a symbol of how fast science responded. Over 13 billion doses have been given worldwide. It was a major moment for global collaboration. For some people, symptoms last for weeks or even months after infection. This is called long COVID. It may be caused by lingering virus, immune overreaction, or reactivation of dormant viruses. Fatigue, brain fog, and chest pain are common symptoms, and it's still a focus of research. SARS-CoV-2 changed how people view science and health. It brought concepts like herd immunity and vaccine development into everyday conversation. Although the virus caused global disruption, it also led to stronger public health systems and better scientific collaboration. Understanding this virus at the microbiological level helps us respond better to future outbreaks. Then I also included a picture of my art piece. Corona means crown in Spanish, and then coronavirus, virus. So she's just the evil queen of the viruses. But yeah, thanks for watching.
I remember watching the 1995 movie Outbreak years ago and being struck by how terrifying it would be to live through something like that. The film depicts a deadly viral outbreak caused by a fictional virus which bears a strong resemblance to the real-life Ebola virus. Watching Outbreak gave me a glimpse into the kind of devastation viruses like Ebola can cause and how fragile our bodies can be in the face of such threats. Ortho-Ebola viruses, named for the Ebola River in Africa, where it was first discovered in 1976, is a highly infectious pathogen known for causing severe hemorrhagic fever in humans and primates, and it's fatal in up to 90% of cases. Ebola belongs to the Filoviridae family, a group of viruses characterized by their filamentous, thread-like shape. Ebola is predominantly found in parts of sub-Saharan Africa and in North Zambia and Angola, covering about 16% of the continent. Among 13 key environmental and social factors studied, temperature, seasonality, and population density strongly influence where the virus can thrive. Specifically, Ebola is more likely to occur in regions where temperatures stay relatively stable throughout the year. There are 5 strains of ortho-Ebola viruses: Zaire, Sudan, Bundibugyo, Tai Forest, and Reston, and they're each named after the location where they were first identified. Zaire, Sudan, and Bundibugyo strains have been associated with major outbreaks in Africa. Over the past decade, there have been 3 major pandemics, including the 2014 Ebola outbreak in West Africa, the 2018 outbreak in Democratic Republic of Congo, and the 2022 outbreak in Uganda. The Reston strain that's found in the Western Pacific is highly pathogenic in non-human primates and pigs, but it is not known to affect humans. Closely related viruses include the Marburg virus, another member of the Filoviridae family, with similarly devastating effects. However, Marburg is less fatal, with only death in about 25% of cases. Ebola is an enveloped filamentous virus and is 80 nanometers in diameter and varies in length up to 14,000 nanometers, often curving back on itself, as you saw in the first slide. Unlike bacteria, viruses like Ebola, they're not cellular. They consist of a protein coat surrounding a small strand of genetic material. In Ebola's case, it's a single-stranded, negative sense RNA genome. And just a quick note on this map image that you see on the slide, you'll see that the Reston strain isn't represented on the map, and I think that's because it's only been known to affect humans to date, but the other 4 are noted on there. Humans are not the natural reservoir for Ebola. The natural reservoir of Ebola is likely fruit bats. Ebola also has a wide host range, and it's capable of infecting diverse mammalian species, including primates, rodents, and bats. Ebola in humans is spread through infected bodily fluids. Some strains of Ebola can spread among monkeys through the air, but aerosol transmission from monkeys to people or among people has not been observed. In humans, Ebola infects and replicates in many different cell types, including monocytes, macrophages, dendritic, and endothelial cells. These interactions affect cellular mechanics and immune processes, which are linked to other important cellular functions, such as metabolism. Macrophage cells get infected first. They overreact by releasing too many signaling chemicals, the cytokines and chemokines. And this causes blood vessels to become leaky and draws more vulnerable cells to the site of infections. So infected macrophages trigger localized blood clotting, which depletes the serum of clotting proteins to an extent that there's just massive uncontrolled bleeding. Endothelial cells don't show obvious signs of damage or destruction when a host is initially infected. Instead, the infection of these cells happens later in the disease during the terminal phase when Ebola is most severe and often leads to death. New Ebola virus particles form inside the host cell and exit via budding. So as new virus particles are assembled, they exit the host cell by pushing through its membrane. And in the process, it takes a piece of the host membrane with it. And this piece already contains viral proteins and becomes the virus's outer envelope. Upon infection, progression of the disease can vary. People with Ebola, they may experience dry symptoms early in the course of illness. These symptoms often include headache, fever, fatigue, dizziness, sore throat, and muscle pain, followed by minor red and purple blood spots or petechia under the skin. As the disease progresses, wet symptoms arise, and the middle stage of Ebola typically includes high fever, massive bloody vomiting, and diarrhea. And late-stage symptoms include damage to blood vessels that cause severe internal hemorrhaging, including bleeding from other body orifices such as the mouth, eyes, ears, and organ damage. And for those who survive an Ebola infection, there are often long-lasting effects, including severe joint pain, eye complications ranging from vision changes to inflammation that can lead to blindness.
Hello friends, my name is Lexi. I am going to be talking about the virus I chose, which is the human papilloma virus, also known as HPV. Starting with an introduction, HPV are small, non-enveloped, epitheliotropic, double-strand DNA viruses that infect the mucosal and cutaneous epithelial cells. This virus causes a variety of proliferating lesions in humans that could be classified as either being benign tumors called papillomas or warts, or being the precursor to cancers and carcinomas by high-risk subtypes. These subtypes can be HPV 16, HPV 18, which are directly related to the development of cervical cancer. HPV is not excluded from other types of cancers associated also with the anogenital and oropharyngeal cancers. Looking at the first box, the virus structure, the HPV virion is a single complete viral particle of HPV. When viewed underneath a microscope, this submicroscopic infection agent can be classified as being spherical shape and measured to approximately 52 to 55 nanometers in diameter. HPV virus is made up of distinctive structures and resemble appearing like a golf ball due to the shape and organization of the caspid proteins, which are L1 and L2. The HPV virion structure is non-enveloped, which means it lacks the lipid membrane surrounding the caspid. It also has a histone structure, which is contained in the caspid protein, as well as a genomic DNA, which is tightly organized structure of circular DNA, which forms a continuous loop. Looking at box 2, I'm going to be talking about the open reading frame. In the HPV genome, an open reading frame, also known as ORF, is a DNA sequence that is potential to be translated into a protein. HPV contains 8 ORFs and transcribed from a single double-stranded DNA molecule. The ORFs are organized in 3 functional regions. These regions are early region, labeled as E, late region, labeled as L, and a large non-coding segment known as the long control region, also known as LCR. These early regions encode proteins E1 through E7, which are essential for viral replication and regulation. The late codes for the structural proteins L1 and L2, which are required for viral caspid assembly. The LCR contains important cis-acting elements that regulate both replication and transcription of the viral genome. Looking at box 3, we're going to be talking about the HPV infection of the epithelial cells. HPV is considered epithelial tropic, which means it primarily infects and replicates within those epithelial cells. It specifically targets the basal layer of the stratified squamous epithelium found at such areas such as the cervix, anus, oropharynx, and skin. Infections typically begin when microabrasions of the minor trauma exposes the basement membrane, which allows for the virus to go down and reach to the basal keratinocytes, the only epithelial cells capable of supporting that HPV replication. The initial binding of the virus is mediated by interactions between the L1 caspid protein and the protoglycans of the host cell surface. The interaction facilitates endocytosis, allowing the virus to enter the cell. Once inside, the viral caspid disassembles and the DNA is transported to the nucleus, where it remains in an episome. A circular DNA molecule separates from the host genome. HPV replication is closely linked to the differentiation of the epithelial cells. In the basal layer, the virus maintains a low copy number of the genome and expresses early genes such as the E1 and E2, which are essential for genome maintenance and replication. As infected cells differentiate and move towards the surface, the virus amplifies its genome expression late genes, which are L1 and L2, which are needed for viral caspid. New viral particles are assembled in the nucleus, released through natural epithelial shedding, avoiding immune detection and not causing the cell to lysis. Looking at box 4, We're going to be talking about the SERV.
Hello everyone, my name is Loy Lu. Today I will talk about one of the most common viruses that affect humans, human rhinovirus or HRV. It's best known as the virus that causes the common cold. What is the human rhinovirus? It's the most frequent cause of the common cold. It's responsible for more than half of all cases. You can catch it by breathing in droplets from someone who coughs or sneezes, or by touching contaminated surface and then touching your face. The virus is active all year, but peak in the early fall and spring. The HRV belongs to the picovirus family. It's very small, just 30 nanometer. It doesn't have an outer envelope, which helps it survive in the environment. One reason HRV is so hard to control is because there are over 160 different types. The virus genetic material is a positive sense RNA strain, about 7,200 base long. This RNA can be directly translated into viral protein once it enters the host cell. To infect a cell, HRV binds to a receptor called ICAM1. Once attached, the virus enters the cell through endocytosis, which means the host cell wraps the virus in a membrane and pulls it inside. Inside the cell, the virus releases RNA, which is translated into one long polyprotein. This polyprotein is then cut into a smaller speed by a viral enzyme. These smaller proteins help the virus copy itself. The new virus particles are made and released either by bursting the host cell or through a process called exocytosis. This entire cycle takes about 8 to 12 hours. The human rhinovirus doesn't just affect a human cell, it also influences other microbes in the respiratory system. It can change the balance of bacteria and other viruses in the nose and throat. HRV might interfere with viruses like influenza A and possibly even reduce the COVID-19 transmission. The virus also triggers the immune response, like the release of the cytokine, which leads to the inflammation. The human rhinovirus only infects humans. It spreads from person to person through respiratory droplets. The virus grows best at a cooler temperature, which is why it prefers the nasal over the lung. The virus can survive on surfaces like door handle and phone for several hours, which makes it easier to spread. For most people, human rhinovirus just cause mild cold symptoms, but for individuals with asthma or chronic obstructive pulmonary disease, also known as COPD, the virus can make their symptoms worse. Early exposure to the virus in children may help their immune system develop and may lower the risk of allergy. In conclusion, human rhinovirus remain a major global health concern. Even though most infections are not serious, the virus has no vaccine and no approved treatment. Research into the human rhinovirus is important not only for finding better ways to prevent cold, but also for improving our understanding of immune system and other respiratory viruses. Thank you for listening.
Respiratory Syncytial Virus Microbiology Spring 2025. RSV is a virus that can affect the upper or lower respiratory system. It causes symptoms similar to the common cold, but can be more severe. Certain populations have a higher risk of experiencing severe infection. These populations include premature infants, people born with chronic lung disease and certain types of heart disease, the elderly, and immunocompromised people. RSV is an enveloped virus that is spherical or rod-like in shape. The surface of the envelope is covered with 3 types of spike proteins. The G protein is an attachment protein that helps a virus attach to human epithelial cells in the respiratory tract. The F protein merges with the human cell membrane so that RSV can empty its contents into the human cell. The SH protein, which stands for small hydrophobic protein, prevents apoptosis of the infected cell and helps the virus replication and survival. Below the membrane, the M protein forms a matrix that links the envelope with the nucleocapsid that contains the genetic information. The genome of RSV is made up of negative-sense single-stranded RNA. It consists of 11 proteins in total and has 10 open reading frames, which are sequences of RNA that can be translated into a protein. RSV is a part of the Paramyxoviridae family and the genus Pneumovirus. It has several relatives. The Paramyxoviridae family includes other viruses that also infect humans, like parainfluenza virus, measles, and mumps. RSV's relatives in the genus Pneumovirus include bovine RSV, which infects cows, and ovine RSV, which infects sheep, along with pneumonia virus of mice. Like all viruses, RSV has no metabolism of its own and instead relies on living cells to replicate and survive. Once RSV has entered the human respiratory tract, it uses the G protein to attach to an epithelial cell. It then merges with the membrane of the human cell and releases its genetic information inside of it. Once inside, the virus takes over transcription and translation to make copies of itself. When the components have been copied, they move toward the membrane where they are assembled into new virions. After assembly, the new virions are released through budding. In the budding process, each new virion uses some of the human cell's membrane to form an envelope that helps it to evade detection by the immune system and go on to infect more cells. The symptoms of RSV are similar to that of the common cold, but may be more severe. Typical symptoms include a cough, runny nose, and congestion. The cough may progress to wheezing after a few days. Some people also experience fever and/or headaches. For some people, especially those in at-risk groups, RSV can progress to a severe infection requiring hospitalization. Patients may need supportive care like oxygen to help them breathe or fluids to help keep them hydrated. Complications of RSV infecting the lower respiratory tract include pneumonia, which is infection of the lungs, and bronchiolitis, which is inflammation of the small airways in the lungs. RSV is spread in the same ways as the common cold. The most common forms of transmission are through direct contact, droplet transmission, and fomites. Direct contact can happen when kissing someone who is sick or touching their contaminated clothing. Droplet transmission happens when we come into contact with the droplets released when a person coughs or sneezes. Fomite transmission happens through the sharing of cups, water bottles, or utensils. Vaccines for RSV first became available in 2023. There are a few different options available. Pregnant people can receive the vaccine during weeks 32 through 36 of their pregnancy. This offers their newborn protection through their antibodies for around 6 months, which should be long enough to get them through their first RSV season when they are most vulnerable. The vaccine is only available during one pregnancy, so future children will need to receive monoclonal antibodies instead. Monoclonal antibodies are available for infants under 8 months old who are in their first RSV season. Certain populations of children between age 8 and 19 months are also eligible for monoclonal antibodies during their second RSV season. Both prenatal vaccines and monoclonal antibodies offer infants passive immunity that only lasts for a few months. Adults over age 60 are also eligible for the vaccine and have a couple of options available. Protein-based vaccines introduce the F protein into the body so that the immune system can learn to mount a defense against it. The mRNA vaccine instructs the body to produce the F protein rather than introducing it directly, so the immune system can learn to recognize and respond to it. Sources for this presentation are cited below.
This comic strip is going to take a look at the hepatitis
B virus and its accompanying interactions. The Adventures of Sully and Friends. Sully and friends visit the virus vet. Outside the clinic, the friends reunite with the lovely virus vet. Hello again, Dr. Bleakley. So, you guys want to learn more information about the hepatitis B virus. Let's take a closer look. Inside the lab, Dr. Bleakley shows the friends some petri dishes. Meet the hepatitis B virus, or HBV. Tiny, sneaky, and bad news for the liver, can cause inflammation and more dangerous diseases. Susie states, "He looks so small, but dangerous." And Dr. Bleakley responds back, "Oh, you have no idea." Let's take a look at its shape and anatomy. Susie excitedly states, "He's got a spiky ball shape. And is that DNA inside?" "Exactly," states Dr. Bleakley. "He's got an envelope with surface antigens to sneak into liver cells." Tony responds, "Like a disguise!" Let's take a look at a virus family tree. HBV is part of the Hepadnavirdae family, viruses with circular, partially double-stranded DNA. Unlike the flu, which is RNA, it uses reverse transcription, kind of like HIV. "Wait," states Tony. "Does this mean that primates can get it too?" Let's take a look inside an infected liver. Dr. Bleakley explains that HBV enters liver cells and uses their own machinery to copy itself, and does so via reverse transcription. Sully questioningly states, "That's weird. Don't DNA viruses usually skip the RNA step?" Dr. Bleakley responds, "HBV, a bit of an oddball." On to page 2. See, you guys aren't the only ones with friends. What is that with HBV? Zooming in, they notice HDV states, "I'm useless without my buddy." Hepatitis D can only affect someone who already has the HBV virus already. Together, they're a lot more dangerous, Dr. Bleakley explains. "So viruses can have toxic relationships too?" says Susie. Since HPV is transmitted through bodily fluids, it's really important to keep in mind hygiene. Remember, says Dr. Bleakley, HPV only infects humans and primates, but it can survive on surfaces for a week. Tony, yikes! So hygiene really does matter. Now let's contrast these two livers. One HBV negative and one HBV positive. Ugh, that looks painful, states Sully. HBV can cause hepatitis, cirrhosis, and even cancer if it sticks around too long. Tony asks, "And some people don't even feel sick?" Dr. Bleakley responds, "Exactly. That's what makes it sneak." And this is why vaccines are so important, Dr. Bleakley explains. Vaccines train the immune system to fight HBV before it attacks. And thanks to research, we know how to prevent it and how to treat it better. "Science saves the day!" exclaims Susie. Dr. Bleakley is now showing you a recap of all of the different infographics as they've taken their journey to understand HBV. Um, and lastly, we have our credits and or our references. Thank you so much.
Hello everyone. Today we'll talk about HIV, which stands for Human Immunodeficiency Virus. HIV is a virus that weakens the immune system. If left untreated, it can lead to a serious condition called AIDS. It is one of the most studied viruses in the world and has affected millions of people globally. HIV is a small round virus about 100 to 120 nanometers wide. It has a soft outer coat called an envelope made of lipids. On the outside, there are proteins called GP120 and GP41 that help it enter human cells. Inside, it has RNA and enzymes like reverse transcriptase, integrase, and protease. HIV belongs to the Retroviridae family and the lentivirus genus. It is a retrovirus, which means it uses RNA as the genetic material and converts it into DNA inside the host cell. Its closest relative is the simian immunodeficiency virus, or SIV, found in monkeys and apes. HIV does not have its own metabolism. It cannot make energy or food. Instead, it takes over the machinery of the host cells to make more copies of itself. HIV grows by infecting CD4 plus T cells, which are part of the human immune system. It uses the host cell's enzymes and materials to produce new viruses. It cannot grow outside of the living host. HIV weakens the immune system. This makes the body more likely to get infection from other microbes, like tuberculosis bacteria, fungi, and viruses that would normally be easy to fight off. HIV spreads from person to person through blood, semen, vaginal fluids, and breast milk. It does not survive well outside the body and cannot be spread by casual contact or air. HIV has a major impact on human health. It causes immune system failure, which can lead to AIDS. HIV has no helpful or symbiotic role. It affects millions of people around the world and has changed how we approach public health and medicine. ART is a combination of medicines that suppress HIV. It prevents the virus from multiplying and lowers the viral load. With proper treatment, HIV can become undetectable and untransmittable. ART does not cure HIV, but it allows people to live normal lifespans. If someone's viral load is undetectable with treatment, they cannot transmit HIV through sex. This idea is supported by science and reduces stigma. U=U campaigns promote medication, adherence, and HIV education. Here are some HIV prevention methods, like condoms, regular testing, not sharing needles, PrEP, daily pill to prevent HIV, PEP, emergency medicine after possible exposure. Here are some HIV testing options. Antibody test, which is most common. Antigen antibody test, early detection. Nucleic acid test, detects RNA. Testing helps with early diagnosis and treatment. It is private and fast. HIV stigma leads to discrimination and fear. It may stop people from getting tested or treated. Education and support are key to reducing stigma. Famous figures like Magic Johnson have helped fight stigma. HIV can hide in places like a brain, lymph nodes, and gut. They're called reservoirs, where the virus is hard to reach. This makes it difficult to fully cure HIV, even with ART. There is no cure for HIV yet, but researchers are working on gene editing, stem cell transplants, which has cured a few rare cases, therapeutic vaccines, broadly neutralizing antibodies. These approaches offer hope for the future. In this presentation, I covered HIV's biology, how it grows, how it spreads, and how it affects other microbes and humans. HIV is more than just a disease. It also has social and environmental impacts. It's a global issue that affects lives, communities, and families. But we have the tools to fight it: knowledge, prevention, and support. Let's stay informed, get tested, and treat others with respect. The more we understand, the more lives we can help. Thank you.
I did my project on herpes simplex virus. Herpes simplex virus has a double-stranded DNA chromosome packed into an icosahedral capsid which surrounds and protects the viral DNA. The capsid is surrounded by different types of virus-encoded proteins collectively called the tegument. It also contains a host-derived lipid bilayer envelope, spike proteins, and is spherical in shape. HSV-1 is primarily transmitted through direct skin to mucosal contact, such as kissing, touching, or airborne droplets, and infects the skin and mucous membranes, including the central nervous system, eyes, mouth, lips, respiratory system, and genitals. It is estimated that about 70% of the world's population is infected with HSV-1. Most HSV infections are asymptomatic or unrecognized, but others can result in blisters on the skin, mouth, lips, eyes, and genitals. HSV-1 blisters primarily appear on the lips and mouth and are commonly referred to as cold sores. HSV-1 is within the family Herpesviridae and is considered an alpha Herpesviridae, meaning it reproduces more quickly than other subfamilies. Other herpes viruses include HSV-2, Epstein-Barr virus, and varicella zoster virus. Research has shown that HSV-1 infection can increase the risk of developing HIV. Like other viruses, HSV-1 has no independent metabolism and instead hijacks the host to get the nutrients and energy it needs. It does this by manipulating the host's metabolic processes. This can be seen in the alteration of glucose and lipid metabolism within the host. HSV-1 infects cells by entering through membrane fusion, replicates its DNA in the nucleus, assembles new viruses, and can establish latency in sensory neurons with the potential for reactivation. After inoculation, there's an incubation period of 4 to 6 days, during which the virus replicates in epithelial cells. A unique characteristic about herpes viruses is their ability to establish latent infections and evade the host's immune defenses. HSV-1 establishes latency in the dorsal root ganglia. Reactivation can be triggered by host immune status, stress response, hormonal fluctuations, etc. Upon exposure, the glycoprotein spikes on the surface of HSV-1 bind to specific receptors on the host cell surface. HSV-1 then enters the host cell by fusion of the envelope with the host cell membrane. The viral DNA and some proteins enter the nucleus through nuclear pores. Portions of the viral DNA inside of the nucleus are transcribed into mRNA and then into proteins which aid in the formation of new virus particles. DNA replication also occurs. These newly assembled virions bud through the inner nuclear membrane, move through the endoplasmic reticulum, and reach the plasma membrane. They gain their envelope from host membranes and exit the cell by fusion, exocytosis, or lysis. Current treatment methods for HSV-1 include antiviral medications, such as acyclovir and foscarnet. However, these drugs do not completely eradicate the virus and are susceptible to drug resistance over time. There is currently no medication or vaccine to prevent or cure HSV-1. Acyclovir, the most commonly prescribed medication for HSV-1, works by preventing DNA synthesis. And these are my sources. Thanks.